These processes that we've been talking about in the cell are very complex, and yet mistakes still can be made. And so we call these mistakes mutations. And so these mistakes can occur during DNA replication or transcription, and they can be caused by environmental factors, and we call these mutagens. Anything in the environment that causes a mutation is regarded as a mutagen. So here's a list of some known mutagens. So UV radiation would be like from your sunlight and when you go tanning. And this would be natural sunlight and fake, so like the tanning beds. X-rays can be mutagens. There's chemicals in cigarette smoke that are mutagens. Benzoyl peroxide is a chemical that's found in like your face washes that can cause mutations. Nitrate and nitrate preservatives are found in like hot dogs and other processed meats. The HPV or human papillomavirus virus or the Helicobacter pylori bacteria, these are infectious agents. And so they carry mutagens, and if you were to get infected with them, they could cause mutations in your cells. So the effects of mutations can either be neutral, meaning that there's it's not good or bad, no effects, and you probably don't even know that this mutation took place. They can be beneficial, so that would be good for you, or they could be harmful mutations, which would be not so good. Mutations that occur in gametes can be passed on to their offspring. So when sperm and egg unite, if that sperm or egg has a mutation, it will be passed on to the offspring. And then the offspring will inherit that mutation, and all of the offspring cell will have that mutation. Mutations that occur in body cells cannot be passed on to offspring and will be confined to just one cell and its daughter cells. So types of mutations could be chromosomal mutations, and these are mutations that change all or a large part of the chromosome. And these are usually pretty serious. We could have missing or extra chromosomes like we do in Down syndrome. In Down syndrome, there's an extra copy of the 21st chromosome. So if we look between these two karyotypes, there's where the split is. If we look at chromosome 21 on the left, we can see that there are 3 copies, whereas chromosomes 21 on the right only has 2. So the one on the left would indicate Down syndrome, and the one on the right would be a normal karyotype. Other things that can happen to chromosomes is, let's say we have our chromosome pair here, and then this tip might break off, and we're going to label the top of it A and B. And what could happen then is that tip could reattach itself, but it could flip it, and now A would be attached to the green part instead of B. And so that's a type of chromosomal mutation as well. You could have 2 chromosomes. I'm going to color code them again with a blue part and a green part, and then its pair would be red and let's say purple. And what could happen is we could switch and go blue and purple could match up together, and then green and red could join together. So all of these cases happen because a chromosome has broken and it reattaches. These types of mutations down here are a lot more dangerous and have pretty serious effects, including up to death. Frameshift mutations are a mutation caused by the deleting or inserting of a base And so this causes all of the codons to be misread. So if we practice this with a sentence, the big dog ate the red cat. Now if we delete the I in big, do we have the same sentence? We start off with the, but then our next three letters would be B, G, and then the D from dog, then O, G, A, T, E, T, H, E, R, E, D, C, A, T, and then nothing. So you can see that when we delete a letter or a base, it changes how the sentence is read. If you look at the DNA sequence in our example down here, you can see our normal sequence, CAG, CCC, ACT, and this is the amino acid sequence that we want. However, if we were to insert this T, it changes the reading frame, and now this is the amino acid sequence that we would get. And so it totally changes how the amino acids are brought to the ribosome. We can also have an insertion that's not a frame shift, because this time instead of only inserting one base, you've inserted three. So you've inserted a whole new amino acid, and instead of only producing 3 amino acids, you actually get 4. So types of mutations. A point mutation is when you change just one or a few bases, and these point mutations get grouped into 3 categories: either silence, missense, or nonsense. A silent mutation is when the mutated codon codes for the same amino acid, so like CAA, and then we change just the one amino acid, the last A, to a G, and we get the same amino acid, so there's no effect. So a mutation happened, but there's no effect, and so this would be one of those neutral effects that we talked about earlier. The second type of point mutation is a missense mutation, and this is when the mutated codon codes for a different amino acid. So you can see this time we've changed that middle base, and so now instead of glutamine, we've now produced proline. And so this could be good, this could be bad, or this could have no effect at all. Then we have nonsense mutations, and these are very serious mutations. And this is when a mutated codon is an early stop codon. And so, for example, in our CAA example, if we change that first base to a U, it actually changes the codon to a stop codon, and it would end up not making the protein that it was supposed to. And these types of mutations are very serious.